Welcome to Genetics, Populations, Evolution, and Ecosystems. In this topic, we will explore how genetic information is inherited, how populations change over time, and how organisms interact with each other and their environment. Genetic diversity and its origins, genes, alleles, genotypes and phenotypes, meiosis and the generation of variation. Genetic diversity refers to the total number of different alleles present within a population's gene pool. One major source of genetic diversity is mutation, which involves random changes to the DNA base sequence. Mutations can occur during DNA replication or due to environmental factors such as radiation or chemicals. While many mutations are neutral or harmful, some create new alleles that may provide a selective advantage under certain environmental conditions. A second major source of genetic diversity is meiosis. During prophase I of meiosis, homologous chromosomes pair up and exchange sections of DNA in a process called crossing over. This recombination produces genetically different gametes. In addition, independent assortment of homologous chromosomes during metaphase I results in many possible combinations of maternal and paternal chromosomes in gametes. Sexual reproduction further increases genetic diversity because it involves the random fusion of genetically different gametes during fertilization. The importance of genetic diversity lies in its role in adaptation and survival. Populations with high genetic diversity are more likely to contain individuals with alleles that confer resistance to disease, tolerance to environmental change, or improved reproductive success. For example, a population with a wide range of alleles may be more resilient to climate change or the introduction of a new predator. In contrast, populations with low genetic diversity, such as those that have experienced a bottleneck or inbreeding, are more vulnerable to extinction. This links directly to conservation biology, where maintaining genetic diversity is a key aim of protecting endangered species. A gene is a length of DNA that codes for a specific polypeptide or functional RNA, and each gene occupies a specific position, known as a locus, on a chromosome. Alleles are different versions of the same gene and arise due to mutations in the DNA sequence. For example, a gene controlling flower color in plants may have one allele for red flowers and another for white flowers. The genotype of an organism refers to the combination of alleles it possesses for a particular gene, such as homozygous dominant, homozygous recessive, or heterozygous. The phenotype, in contrast, is the observable characteristic that results from the interaction between the genotype and the environment. For instance, two organisms with the same genotype may show slightly different phenotypes if they develop in different environmental conditions. Dominant alleles are expressed in the phenotype even when only one copy is present, while recessive alleles are only expressed when two copies are present. However, not all inheritance follows simple dominance. Codominance, such as in the ABO blood group system, and incomplete dominance also contribute to genetic variation within populations. Meiosis is a type of cell division that produces genetically varied haploid gametes. It consists of two divisions, meiosis I and meiosis II, following a single round of DNA replication. During prophase I, homologous chromosomes pair up to form bivalents and undergo crossing over, where sections of DNA are exchanged between non-sister chromatids. This creates new combinations of alleles on each chromosome. Another important source of variation during meiosis is independent assortment. At metaphase I, homologous chromosome pairs line up randomly along the equator of the cell, meaning that the maternal and paternal chromosomes are distributed independently into daughter cells. This results in a vast number of possible genetic combinations in gametes. Meiosis II then separates sister chromatids, similar to mitosis, further ensuring that each gamete is genetically unique. The significance of meiosis lies in its contribution to genetic diversity within populations. By producing varied gametes, meiosis increases the likelihood that some offspring will possess advantageous traits that improve survival or reproductive success. This variation is essential for natural selection to operate. Without meiosis and the variation it generates, populations would be genetically uniform and unable to adapt to changing environments. Therefore, meiosis provides a crucial link between molecular genetics and large-scale evolutionary processes. Populations, gene pools, and allele frequencies, the Hardy-Weinberg principle, factors affecting allele frequencies. A population is defined as a group of organisms of the same species living in the same area at the same time and capable of interbreeding. The gene pool of a population includes all the alleles of all the genes present in that population. Studying populations rather than individuals allows biologists to understand how genetic variation changes over time. Allele frequency refers to how common a particular allele is within the gene pool and is usually expressed as a proportion or percentage. Several factors can change allele frequencies in a population. Mutation introduces new alleles, while natural selection can increase or decrease the frequency of alleles depending on whether they confer a survival or reproductive advantage. Genetic drift, which is random change in allele frequencies, has a greater effect in small populations and can lead to the loss of alleles entirely. Gene flow, caused by migration of individuals between populations, can introduce new alleles or alter existing frequencies. Understanding gene pools and allele frequencies is essential for explaining evolution at the population level. Evolution is defined as a change in allele frequencies in a population over time. By tracking these changes, biologists can investigate how populations respond to environmental pressures and how new species may eventually arise. The Hardy-Weinberg principle provides a mathematical model that predicts how allele and genotype frequencies remain constant in a population, provided certain conditions are met. Students use the equation p squared plus 2pq plus q squared equals 1, where p represents the frequency of the dominant allele and q represents the frequency of the recessive allele. The principle assumes no mutation, no migration, no natural selection, random mating, and a large population size. If these conditions are met, allele frequencies will not change between generations, and the population is said to be in genetic equilibrium. The value of the Hardy-Weinberg principle lies in its use as a null hypothesis. By comparing observed genotype frequencies with those predicted by the equation, biologists can determine whether evolutionary forces are acting on a population. For example, if the observed frequency of a recessive phenotype is higher than expected, this may indicate selection in favor of that allele or non-random mating. The principle is widely used in conservation biology to assess genetic stability in populations and in medical genetics to estimate carrier frequencies of inherited diseases. Allele frequencies within populations can be altered by several evolutionary factors. Natural selection is a major factor, as individuals with advantageous alleles are more likely to survive and reproduce, passing those alleles on to the next generation. Mutation introduces new alleles, although mutation rates are generally low and usually have a small immediate effect on allele frequencies. Genetic drift causes random changes in allele frequencies and is particularly significant in small populations, such as those that have undergone a bottleneck or founder effect. Migration, or gene flow, occurs when individuals move between populations and interbreed, potentially introducing new alleles or changing existing frequencies. Non-random mating, including inbreeding and sexual selection, can also affect genotype frequencies and indirectly influence allele frequencies. Understanding these factors allows biologists to explain why real populations rarely remain in Hardy-Weinberg equilibrium. Together, these processes drive evolutionary change and help explain patterns of variation and adaptation seen in nature. Natural selection as a mechanism for evolution, speciation and reproductive isolation, classification, phylogeny and evidence for evolution. Natural selection is the primary mechanism of evolution and occurs because individuals within a population show variation in their characteristics, much of which is genetically determined. Environmental factors, known as selection pressures, mean that some individuals are more likely to survive and reproduce than others. Those with advantageous alleles are more likely to pass these alleles on to the next generation. Over time, this leads to an increase in the frequency of advantageous alleles in the population. Natural selection can result in different types of selection. Directional selection favors one extreme phenotype, such as antibiotic resistance in bacteria. Stabilizing selection favors intermediate phenotypes and reduces variation, for example in human birth weight. Disruptive selection favors both extremes and can increase variation within a population. Natural selection links genetic variation to environmental conditions and explains how populations become better adapted to their surroundings. It also provides a foundation for understanding speciation and biodiversity. Speciation is the process by which new species are formed, and it occurs when populations become reproductively isolated. In allopatric speciation, a physical barrier such as a river or mountain range separates a population. Over time, different selection pressures and genetic drift cause the populations to diverge genetically. Eventually, they become unable to interbreed even if the barrier is removed. Sympatric speciation occurs without geographical separation and may result from reproductive isolation due to behavioral differences, ecological niches, or polyploidy in plants. Reproductive isolation can be prezygotic, such as differences in mating behavior or timing, or postzygotic, where hybrid offspring are infertile or non-viable. Speciation explains the diversity of life on Earth and shows how evolution can lead to the formation of distinct species from a common ancestor. This topic integrates genetics, selection, and population biology. Classification is the systematic arrangement of organisms into groups based on shared characteristics, and it reflects evolutionary relationships. Modern classification systems are based on phylogeny, which shows how species are related through common ancestry. Evidence for evolution includes fossil records, which show changes in organisms over time, and molecular evidence such as comparisons of DNA and amino acid sequences. The more similar the sequences, the more closely related the species are likely to be. Homologous structures, such as the pentadactyl limb in vertebrates, also provide evidence of common ancestry. Classification is important for communication, conservation, and understanding biodiversity. Ecosystems, habitats and ecological niches, energy flow and trophic levels, nutrient cycles in ecosystems. An ecosystem is defined as a dynamic system made up of a biological community and its physical environment. Within ecosystems, organisms occupy specific habitats and ecological niches. A habitat is the place where an organism lives, while a niche describes the role it plays in the ecosystem, including its interactions with other organisms and the environment. Niches include factors such as feeding relationships, activity patterns, and tolerance ranges for abiotic conditions. Two species cannot occupy exactly the same niche in the same habitat for long, a principle known as competitive exclusion. Understanding niches helps explain species distribution, competition, and biodiversity. Energy flows through ecosystems in one direction, from the sun to producers and then through consumers and decomposers. Producers, such as green plants, convert light energy into chemical energy through photosynthesis. This energy is transferred between trophic levels through feeding. However, energy transfer is inefficient, with significant losses at each level due to respiration, movement, and waste. Typically, only about 10% of energy is transferred to the next trophic level. This explains why food chains are usually short and why there are fewer organisms at higher trophic levels. Energy pyramids are used to represent this decrease in available energy. Understanding energy flow is essential for explaining ecosystem structure and the impact of human activities on food webs. Nutrient cycles describe how essential elements such as carbon and nitrogen are recycled within ecosystems. The carbon cycle involves processes such as photosynthesis, respiration, decomposition, and combustion, which move carbon between the atmosphere, organisms, and the environment. The nitrogen cycle includes nitrogen fixation by bacteria, nitrification, assimilation by plants, and denitrification. These cycles ensure that nutrients remain available for biological processes. Human activities, such as fossil fuel combustion and intensive agriculture, can disrupt nutrient cycles, leading to issues like climate change and eutrophication. Ecological succession, measuring and maintaining biodiversity, conservation strategies, and human impact. Ecological succession is the process by which ecosystems change over time. Primary succession occurs on bare land where no soil is present, such as after a volcanic eruption. Pioneer species, such as lichens, colonize the area and gradually create soil, allowing other species to establish. Secondary succession occurs in areas where disturbance has removed organisms but left the soil intact, such as after a fire. Succession leads to changes in species composition, biomass, and biodiversity. Over time, a stable climax community may develop, although modern ecology recognizes that ecosystems are dynamic rather than static. Succession demonstrates how ecosystems develop and recover, linking ecological processes to long-term environmental change. Biodiversity can be measured at the genetic, species, and ecosystem levels. Biodiversity is often quantified using species richness and indices such as Simpson's Index of Diversity. High biodiversity is generally associated with ecosystem stability and resilience. Maintaining biodiversity is important for ecological, economic, and ethical reasons. Diverse ecosystems are better able to withstand environmental change and provide resources such as food, medicines, and ecosystem services. Measuring biodiversity also supports conservation planning by identifying areas or species at risk. Practical skills such as sampling using quadrats and transects are essential for collecting biodiversity data accurately. Conservation aims to protect biodiversity and prevent extinction. In situ conservation involves protecting species within their natural habitats, such as nature reserves and national parks. Ex situ conservation includes methods like zoos, seed banks, and captive breeding programs. Each approach has advantages and limitations. Human activities, including habitat destruction, pollution, overexploitation, and climate change, are major threats to biodiversity. Understanding these impacts allows scientists and policymakers to develop effective conservation strategies. In summary, genetics explains inheritance and variation, populations provide the basis for evolutionary change, and ecosystems demonstrate the interconnected nature of life on Earth. Together, these concepts help us understand the diversity of living organisms. Thank you for watching.